To Grand Rounds today. Today's a special Grand Rounds because we've invited a speaker from outside, from Johns Hopkins, Sidney Kimmel Cancer Center, Dr. Nilofer Azad. Dr. Azad is Associate Professor of Oncology and a member of the Gastrointestinal Oncology Program at Sidney Kimmel. She completed her fellowship in medical oncology at the NCI and became a member of the faculty there in 2008. Since joining the faculty at Sidney Kimmel at Johns Hopkins, Dr. Azad is the PI of numerous early phase clinical trials in solid tumors and gastrointestinal tumors. That's how many of us know her through collaborations. Dr. Azad is a clinically active medical oncologist and is a co-leader of the NCI-funded UM1 Developmental Therapeutics Clinical Research Program at the SKCCC. We have one of those here. Pat LaRusso is unfortunately in Washington today, but she sends her best. She's a member of both the Epigenetics and colon cancer stand-up to cancer dream teams, serving as a principal on the latter. She's a member of the NCI Colon Cancer Task Force, the advisory board on the Biden Cancer Initiative, and the executive board on the Cholangiocarcinoma Foundation, among others. She has a laboratory dedicated to translational research and drug development that she uses to inform her clinical trials. Research, as we'll hear about today, is focused on epigenetic alterations in cancer cells, changes in gene expression due to potentially reversible modifications of DNA, and I'll let her tell us more about that. But it's really wonderful to have Nilofer here, someone who really takes cancer therapy from the bench to the bedside and back, and we're so grateful that you've taken time to visit us, and we look forward to your talk. Thank you.
Well, thank you, Roy, for that kind introduction. And thank you for the invitation to be here today. It's a real honor for me to be here today to talk about this work that we've been working on in our group for the last 10 years or so, which is how can we move epigenetic therapy forward in patients with solid tumors, especially with GI cancers. And it's particularly special for me because your relatively new chairman of surgery, Nita Ahuja, is really the person responsible for bringing me into this field a decade ago when I was a junior faculty member. I had just started at Hopkins, and Nita was already a world-renowned epigeneticist and wanted to have a collaborator in medical oncology. And so she reached out to me and has really mentored me over these last many years. So it's very special for me to be able to present the work that our group has been working on for all these years. And there's a lot of reason for us to be enthusiastic, those of us in the epigenetics community, about the possibility of using epigenetic modulators in cancer therapy. Because over the last 20 years, if you think of what we have learned about cancer, underlying cancer biology and abnormalities, we've understood and been able to profile a litany of genetic mutations and fusions, abnormalities, and we now, for most common cancers and even uncommon cancers, can describe in detail the genetic abnormalities that are there, but we can't correct any of these abnormalities. So we've got targeted therapies that deal with the ramifications of a given genetic abnormality, but when it comes to correcting the underlying lesion, we really can't change that underlying lesion. And that is completely in opposition to what we can do with epigenetic abnormalities. We also have profiled and found that a given cancer generally has hundreds of epigenetic abnormalities as well, but those are abnormalities that with epigenetic modulators we really can reverse. And it gives us the potential of possibly changing the underlying biology of a cancer to maybe make it more immunosensitive or more chemosensitive, and that's really the area that we're going to talk about today. When we talk about the two different groups of epigenetic modulators today that I'm going to discuss, we're really going to focus on drugs that focus on post-translational modifications of histones, especially histone acetylation, which makes a major difference in how chromatin is configured, and then DNA methylation, which can happen both in normal cells and abnormal cells, as we know, to silence gene expression. And this is a very simplified cartoon of why histone acetylation and DNA methylation matters when it comes to gene expression. So if you take an area of the genome, especially if you're looking at gene promoter regions, and you've got areas of the gene promoter region that are methylated, which is represented by the red circles here, or you've got unacetylated histones, you're going to have a more tightly packed down chromatin configuration that's going to impede transcription, and the DNA methyl groups on those promoter regions are also going to impede the binding of transcription factors. The opposite is true when you have areas of chromatin that are open, that are with histones that are acylated, and with DNA that is demethylated in these gene promoter regions. Now, there's much complexity to epigenetic modulation. I'm narrowing it down to these two components because that's where we've had the most information and the most preclinical and clinical data when it comes to using these kind of drugs. So DNA methyltransferase inhibitors, as we all know, are FDA approved in myelodysplasia and AML. Decitabine and azacitidine are both approved in that setting. These have very short half-lives though. So when it comes to solid tumors, there's a real challenge in terms of being able to have a pharmacodynamic effect at the tumor level when you've got a drug that is so quickly broken down in the circulation. And that's why in the solid tumor community, I just met with Steve Gore earlier today who said that he thought that guadicitabine was crap, as he said. But guadicitabine, which has a more prolonged half-life, in solid tumors may be more important because we really need time to have these drugs get to the tumor. And guadicitabine is a decitabine prodrug, and we have now many different trials where we have had tumor biopsies in these patients and shown that we actually can get good demethylation with these new generation demethylating agents. Now, histone deacetylase inhibitors have been in the clinic for 20 years. There is some fair skepticism, I would say, about the ability to use HDAC inhibitors in solid tumors. They are FDA approved in cutaneous T-cell lymphomas, but I'm going to show you, I hope, a wealth of data that has been generated, especially in the last 5 years, that indicate that we might have ways we can really use these drugs in solid tumor patients as well. But I will say from the outset that multiple clinical trials looking at epigenetic agents alone have been negative in every solid tumor that they've been tested in. And I'm specifically talking about DNA methyltransferase inhibitors and HDAC inhibitors, but we've looked at them alone and we've looked at them together with each other. And that effort, looking at HDAC plus DNA methyltransferase inhibitors alone, was really led by the initial Stand Up to Cancer Dream Team. Steve Balin and Peter Jones were the head of that. That's when I came into the epigenetics community. We were running 3 clinical trials in colon, breast, and lung cancer, and all 3 of those trials were negative. And so the use of these agents as single agents in solid tumors is not appropriate and doesn't work. But there are some small subsets of patients that are genomically defined where epigenetic modulators might be effective. So bromodomain inhibitors have been shown to have activity in nut midline tumors which have a BRD4 mutation. And then I put a question mark here in terms of IDH1 mutated cancers because IDH1 mutated cholangiocarcinoma, we are seeing some benefit with IDH1 inhibition. But it's a little bit controversial whether IDH1 is really considered an epigenetic modulator or not. IDH1 is, as you all know, an important gene and protein that's involved in aerobic glycolysis. In the setting of having a mutation of IDH1 and 2, we have a buildup of 2-HG, and this leads to widespread epigenetic dysregulation. And so even though IDH1 inhibition is not necessarily a pure epigenetic modulator, it has dramatic epigenetic impact, which has been shown in many preclinical studies. This is intrahepatic cholangiocarcinoma because it's a GI cancer and that this is an area where this actually is functional and clinically beneficial. I did just want to highlight it with a couple of slides. 25% of intrahepatic cholangiocarcinoma has a mutation in IDH1 or IDH2, predominantly IDH1. And the initial phase 1 study of which we were a part did show that even though there were very few RECIST criteria responses in these studies, that a majority of patients at least had stable disease, with 40% of those patients having continued stable disease even at 6 months. We've just had this paper accepted to Lancet Gastroenterology, which expands on that with 73 patients that were treated with intrahepatic cholangiocarcinoma, showing, again, a small response rate of 5%, but a broad number of patients having stable disease. And you can see here on the swimmer's plot that, again, around 40% even in the expanded cohort have 6 months or greater progression-free survival. But that's really it when it comes to solid tumors and single agent activity of these kinds of drugs. And where the money really feels like it is in combination therapies, especially to change chemosensitivity and immune sensitivity. And I'll talk about chemosensitivity first. I just want to highlight the initial dream team that I was talking about, because this has really been a collaborative effort across different groups of basic science investigators and laboratory investigators to get this field to where it is now. So I'll go back to the trials that I mentioned a few moments ago. We did 3 clinical trials in heavily pretreated lung cancer, colon cancer, breast cancer. All 3 of those trials used the same doses of azacitidine and entinostat that had been previously defined in other clinical trials. Many of those trials that Steve Gore was actually involved in when they were run as phase 1, and all 3 were negative. But luckily, John Rangel, who was a fellow at that time at Hopkins, decided to do a post hoc analysis to look at what happened to the lung cancer patients when they went on to their subsequent therapy. And this is the analysis that John did. And what he found was in this heavily pretreated group of lung cancer patients, so these are 4 or more prior therapies as a median, 30% of those patients went on to have a RECIST criteria response. And another 30% or more of patients went on to have stable disease. So this is really not what you would expect for heavily pretreated lung cancer. And it just began the question about whether or not we were seeing some sort of a priming effect or whether this was just happenstance. So other groups had looked at this. In GI cancers, this had only really been explored in a couple of studies, one preclinical and one clinical with oxaliplatin, showing maybe some priming with oxaliplatin in vitro-based studies, as well as xenograft studies. But when the study was performed at MD Anderson, looking in patients across GI tumors, that trial was completely negative. But in breast cancer, the use of HDAC inhibitors was looking and is looking more compelling. And even though today we're talking about GI cancers, I do want to highlight a few places where we really might have some opportunity to use these agents in solid tumors. In breast cancer with resensitizing to hormonal therapy, and then in ovarian cancer looking to resensitize to platinum therapy. So this is the breast cancer preclinical data, initially looking at entinostat in an animal model system and showing what happens as this tumor initially is controlled by letrozole, and then entinostat is added, and we get benefit when entinostat is added, even if you continue letrozole or if you switch to exemestane, when none of these have any benefit if they're continued as a single agent. So ENCORE 301 was a clinical trial that reported on a randomized phase 2 trial of this combination, looking at exemestane and entinostat versus exemestane alone. So these are all in patients that had previously progressed on an aromatase inhibitor. And the p-value that they were looking at for a one-sided significance level was 0.1. So this is a double-blind placebo-controlled study, and they did meet their one-sided p-value of 0.05 with a 2-month improvement in progression-free survival. But what was surprising was that the overall survival benefit was substantially greater. So there was an 8-month statistically significant survival in overall survival for the entinostat exemestane treated patients, suggesting that potentially entinostat had better effects even after the patients progressed on the exemestane therapy. And this has led to a large cooperative group study that is presently completing enrollment. This is a trial, again, looking at patients who had previously been treated with an aromatase inhibitor and randomizing these patients to exemestane plus entinostat versus placebo. The sample size is increased for an overall survival endpoint. And this is a trial that's being run by my recent former colleague, Rasheen Connolly, who's just left Hopkins to move back to Ireland. But we hope to see these results sometime soon. And this is the best chance that we have in terms of a phase 3 trial to potentially result in having an HDAC inhibitor approved in a solid tumor. Now, I mentioned chemosensitization as well. In ovarian cancer, that data has gone the furthest. This is preclinical work looking at SGI-110 or guadocytabine, the next generation demethylating drug I mentioned, and showing here that in ovarian cancer animal models, platinum-resistant disease became more sensitive, significantly more sensitive when the demethylating agent was added, even though there was almost no activity of the demethylating agent alone. The initial clinical trials that they did was using decitabine, IV decitabine, showing some responses in platinum-resistant disease with a progression-free survival in these patients of 8 months, but this was a non-randomized trial. And so the group in this all of this work has really been led by Daniela Mattei and Ken Nephews out of Indiana, Daniela now being at Northwestern, but they ran a randomized phase 2 study of 100 patients and found that there was a progression-free survival benefit for the guadocytabine plus platinum treated patients, 16 weeks versus 9 weeks, with a PFS at 6 months that was also improved, and a somewhat improved overall survival that did not meet statistical significance in this trial. So Nita and I were interested in exploring whether this was happening in our colon cancer patients after they came off of our epigenetics trial as well. But unfortunately, this was back in 2013, 2014, and at that time, our patients really didn't have a subsequent therapy to go on to. All of our patients had been prior treated with oxaliplatin and irinotecan, 5-FU, and so most of our patients actually moved on to comfort care after they came off of the study. So Nita and her lab, and this is work that was led by Anup Sharma, who is now here at Yale, moved forward in looking at this in the laboratory. So Anup's work first tried to define a dose of a demethylating agent that would not be cytotoxic in order to really focus on the demethylation component of the demethylating agent rather than a cytotoxic effect. And defining this lower dose of azacitidine, they were able to show here that they were able to decrease DNA methyltransferase 1, and down here in panel E, what you'll see is across a panel of cell lines, they were able to show global demethylation that was significantly greater with azacitidine than they saw for MOC. Anup then profiled these same cell lines treated with azacitidine against a screen of multiple chemotherapy drugs and found that of those chemotherapy drugs, irinotecan was the one where there was the best chance of having a sensitization effect, and moved forward looking at first in vitro assays and showing that in cell lines like HCT116, which is extraordinarily sensitive to irinotecan, there was really no additional benefit of adding a low dose of azacitidine, but in resistant cell lines, there was a benefit to adding these low doses of azacitidine, similar in clonogenic assays. And then here in vivo assays, again, if a cell line was already sensitive to irinotecan, there was no additional benefit. This is the pink line that is irinotecan alone versus the blue line, that's the combination. No benefit to adding azacitidine to those animals. On the other hand, in a very resistant cell line, as you can see, both azacitidine and irinotecan having no effect, but when you combine the agents, you get complete abrogation of tumor growth. So that was exciting, and we thought that that was worth moving forward into clinical trials, and so we did. We moved this forward into a clinical study combining guadocytabine with irinotecan in patients that had metastatic colon cancer. And this clinical trial was a study that was designed, it was a phase 1 study, so it had a standard 3+3 design, but every patient had to have prior exposure to irinotecan before they were allowed to go on this study. And we incorporated paired tumor biopsies so that we could later, after the trial was completed, do some correlative work to see if we were really seeing demethylation or not, and if there was something predictive in the biopsies that would be helpful. So we started our trial and immediately found that even at doses that were substantially lower than what was given in the MDS trials, we had significant toxicity in terms of neutropenia and neutropenic complications. And so we ended up having to de-escalate and add growth factor support. And with the addition of growth factor support, we were able to re-escalate and get back up to a dose level that we were interested in being at, where we knew that there had previously been signs of demethylation in other clinical trials. This is our table 1, which just shows that this was a very standard colon cancer patient population. It's a small study because of phase 1 of 22 patients, but all the patients had prior exposure to the agents that you would expect in a phase 1 study focusing on colon cancer, and these patients had good performance status. And this just shows the dose-limiting toxicities that we saw in these patients, really all focused on neutropenic fever and neutropenic complications, but really decreasing substantially once we added growth factor support. But you'll also see here in terms of the number of cycles that we had many patients that even with prior exposure to irinotecan were able to remain on this treatment and tolerably remain on this treatment for a significant period of time. Our overall toxicity profile was what we expected when we combined these agents, so significant issues with leukopenia and neutropenia, but overall, otherwise, we were seeing the toxicities that we would expect with irinotecan, predominantly low-grade GI toxicities, as well as some anorexia as well. But again, what we found interesting was that in a subset of these patients, about half of the patients, we were seeing durable benefit in terms of at least stable disease in these patients. And biochemically, we were seeing similar effects in terms of our tumor markers as well. And I always like to highlight this particular patient. She was a patient of Nita and mine. We shared her. Nita performed all of her surgeries, and I took care of her in medical oncology. But this is a case that's really illustrative of how epigenetic therapy may really be changing patients' underlying biology. So this patient, Trina, came to us as a 42-year-old who had really high-risk stage 3 disease. She underwent adjuvant chemotherapy with FOLFOX, and the first time that she got restaged, 1 month after she completed adjuvant therapy, she already had recurred. So anyone who takes care of colon cancer can tell you that that is a harbinger of badness for a patient. She then got chemoradiation to that area that recurred, and immediately, next scan, she progresses and has progressive disease with peritoneal metastasis. We give her FOLFIRI, she goes to surgery, she comes off of FOLFIRI, and immediately progresses again. She was enrolled on a clinical trial of a MUC5 inhibitor because she had MUC5 staining. This was a study that was running through our phase 1 program. Immediately progressed on that study as well. So every therapy that she had, she either progressed on or she progressed as soon as that therapy ended if it was in the adjuvant setting, until she came on this trial of glucosidobine and irinotecan and was able to stay on this study for almost 2 years and did very well on it. And this is her scans. You can see this very thick rind of tumor that was present that basically disappeared within a few months on therapy. And this stayed that way for her, as well as her changes in CEA for all of the time that she was on study. In fact, when she came off of study because of a single liver lesion that was elsewhere that had come up as a new lesion, which met RECIST criteria for progressive disease. So our clinical results of this study showed that our patients were able to be on study for about 4.5 months, and that historical control to that is regorafenib, Stivarga, or Lonsurf, where the median progression-free survival was 1.9 months or 2 months. Now, this is a much smaller study and run at only one institution, so that's just a historical control, but we thought that it was interesting enough to follow up on. We did see benefit at both the 30 and 45 milligram cohorts, and then we decided that we wanted to use our correlative specimens to help define whether we should be aiming for a higher or lower dose level between these two doses. And so we, as I mentioned, had timed our correlates to try to make it as perfect as possible to only look at the effects of glucosidobine on the tumor without any compelling contributing factor of irinotecan. So we added a biopsy after only 8 days on therapy. And this is how the perfect can really get in the way of what is good, because only 8 days of therapy with a demethylating agent was, in hindsight, a mistake. And what we did was assess this using line 1. So line 1 is an assay that is used as a surrogate for global demethylation. It looks at certain areas of line elements in tumor DNA. About 17% of our genome is made up of line elements, and these are highly methylated areas. So it's a surrogate of global demethylation, but it was also the assay that was used in the initial studies of glucosidobine in MDS AML. And what this curve here shows from that figure from Jean-Pierre Issa's work is that when you got to 60 milligrams per meter squared, there was really no increase in terms of demethylation. And even at 45 milligrams or 36 milligrams per meter squared, we were seeing significant demethylation in MDS. So we really thought that this was kind of the sweet spot that we wanted to be in. And we looked at initially our tumor biopsies, and what we found in this is that there was very variable. So when you look at these tumor samples at day 8, the demethylation was really all over the place. But when we went and looked at our serial blood samples, both looking at WBCs as well as looking in circulating tumor DNA, by cycle 2, we were seeing consistent demethylation across the samples. And that we were seeing demethylation at both the 30 and the 45 milligram per meter squared dose, though there did seem to be more depth to that demethylation at the 45 milligram dose. And so that is what supported us moving forward with that dose level, but feeling comfortable about a dose reduction for the patients that needed it down to 30 as well. And that's how our phase 2 study was designed, with a 45 milligram dose level based on correlative work. And I just say this because we often get pushback in phase 1 studies to incorporate endpoints and correlative endpoints unless we can demonstrate at the beginning what we are absolutely going to do with it. But being able to have those specimens really helps inform future clinical trials, even though that wasn't the initial intention of those biopsies. So this was a randomized phase 2 study looking at irinotecan and guadocytabine versus regorafenib or Stivarga as our control arm. It's a 2-to-1 randomization, and all of these patients have to be irinotecan resistant. And using these data, we also moved the biopsies for these patients into cycle 2 so that hopefully we'll have more information from this clinical trial. This study is completely accrued and is presently undergoing analysis, and I'm hoping that we will have these data to present to you all by ASCO in January, ASCO GI. So the next major area that we've been interested in moving epigenetic therapy forward is in strategies that look at immunomodulatory combinations. And again, I'm going to take you back to this figure that I showed you a few moments ago, where when this analysis was done, this post hoc analysis of the lung cancer patients, what was interesting is it was completely accidental, but it turned out that Julie Bramer was running the very first studies of the Medarex drug that we now know as nivolumab. That drug was in phase 1 at Hopkins. And so a few patients from the epigenetic trial progressed on their epigenetic therapy and happened to go on to their next trial, which was the PD-1 inhibitor. And every one of those patients, it was 6 patients, but every one of those patients did well. And 3 of those patients had very deep, durable responses to therapy. That's these patients here. Even the patients that didn't have a RECIST criteria response were on study for over 6 months without progression. And so it's difficult in a setting where you've got a drug that you know now is active in lung cancer to know whether this was just the fact that some patients were going to do well on the PD-1 inhibitor and this had nothing to do with the fact that the patient had epigenetic therapy first, or whether this was really a priming effect from the epigenetic agents. But what I will posit to you is that if you look at what a cancer cell has to do in order to survive the immune system, it makes an inordinate amount of sense that you might want to combine it with an epigenetic agent. And I still think this is a fantastic figure from the science paper from Schreiber, looking at and trying to detail what happens with a tumor that is able to be eradicated by the immune system versus one that's able to escape the immune system. And if you look at the things that you might want in a tumor microenvironment in order to have a good immune response, if you want to lower the number of immunosuppressive cells in that tumor microenvironment or increase the number of cytotoxic cells, if you want to increase immune checkpoint expression on tumors or on immune cells, all of these things, we've got data that support epigenetic therapy can do these things. It can increase MHC class expression. And in the next few minutes that we have together, I'm going to try to talk you through some of the data that exists, but these are just representative papers and studies that have been done that have shown this, but there have been many reports in the last 5 years that have really focused on the immune modulatory effect of epigenetic therapy. So this is hopefully going to give you a flavor of those data. So if we first want to look at tumor-associated antigen expression, there's a running hypothesis that tumor-associated antigens or neoantigen expression may be responsible for responses to immunotherapy. These are tumor-associated antigens. On the left, you see this in prostate cancer. On the right, in Mike Topper's Cell paper in lung cancer. And they've used a heat map format to show increases in gene expression across these tumor-associated antigens with demethylating agents, HDAC inhibitors, or the combination. And this has also been demonstrated by Nita's group in colon cancer. It's been demonstrated by other groups in breast cancer, ovarian cancer. So this is a perpetual theme that's been shown by many groups that we can increase antigen expression. When we look at co-stimulatory molecule expression that's necessary for an immune response, necessary but not necessarily sufficient. We do have increases that have been shown in MHC class molecules and CD40, CD80, HLAs, increasing with exposure on the left here to HDAC inhibition, on the right to both HDAC inhibition and demethylating agents. This here in melanoma. These are some of the earlier studies, but there have been other studies that have come since here too. And I also think, interestingly, if you look at the cytokine profile that happens after exposure to epigenetic modulators, there's also a shift in vitro and in vivo in the cytokine profiles to a more pro-inflammatory cytokine profile, with increases in IL-2 and interferon gamma, decreases in IL-10. And in the heme literature, patients that have been treated with azacitidine, this has also been demonstrated in patient samples as well, in terms of this shift in cytokine profile as well. And then in just the last 2 years, there have been multiple papers that have also looked at immune checkpoint expression with epigenetic modulators. Here in Pam Munster's work, looking at various epigenetic modulators in breast cancer and showing increased PD-1 expression across breast cancer cell lines. She's also showed this in primary xenografts in another paper. Here on the right, in ovarian cancer models, looking at OX40 and 4-1BBL expression increasing both with pharmacologic as well as siRNA-based inhibition of HDAC as well as DNA methyltransferase. John Rangel, who is the fellow I had mentioned previously, he's now a faculty member in South Carolina, had also shown PD-L1 expression increases in lung cancer cell lines. But John also then did an analysis looking at gene sets and pathways that were upregulated when cell lines were exposed to azacitidine, and showed that across the majority of lung cancer cell lines, there was significant increase in immune-related pathways. Mike Topper has taken that work forward and tried to refine it even further, looking at and again showing that there are many important gene sets involved in the inflammasome and the immune pathways that are upregulated, but also showing that a given cell has unique pathways that are upregulated compared to another given cell line, and that there is some commonality, but there's also pathways that are unique to each individual cell line and likely to each individual tumor as well. Anita will recognize this paper that also really focused on gene set pathway analysis. With Cindy Zanow, their work really focused on trying to identify those commonly upregulated gene set pathways. And so this was in a large panel of breast, ovarian, and colon cancer cell lines that were treated with low doses of azacitidine, and then the common pathways that were up and down regulated were assessed, and they found that immune-related pathways were a common subset of pathways that were upregulated. Using these pathways, they derived a signature of azacitidine exposure that leads to immune-related activation, and went to the TCGA and found that if you looked at TCGA primary tumor samples, so these are all untreated with epigenetic therapy, right? So primary tumor samples can still segregate and cluster based on this immune gene signature. And those of us who did colon cancer particularly found it interesting that in colon cancer, the patients that had the high immune gene cell signature from azacitidine, but they had it de novo, were the microsatellite unstable cancers, which we now know are highly sensitive to immunotherapy. And so that was biologically a satisfying output when these samples were assessed. Now, other groups have really tried to focus on what happens in the tumor microenvironment in terms of which cell types are upregulated or increased or decreased with treatment with epigenetic modulators. So this is work looking at SAHA or vorinostat, which is an HDAC inhibitor in a peritoneal mesothelioma model, first staining for CD8 positive cells and showing that in this animal model, CD8 positive cells increased with exposure to HDAC inhibition, while regulatory T cells, such as shown here, decreased. So both going in the direction that you would want for an improved immune response. Roberto Pilli has really focused on this extensively in terms of his interest looking at the impact of HDAC inhibitors in renal cell cancer. And so Roberto first showing in the RENCO2 model that the known compensatory increase in regulatory T cells that happens with IL-2 therapy is able to be abated with the use of entinostat. This does also potentially result in improved benefit when these agents, entinostat in particular, is combined with PD-1 therapy. Roberto has taken this into the clinic, and so this is his report of his phase 1 study looking at entinostat in combination with IL-2 and showing, so this is in untreated patients with renal cell cancer, but had an almost 40% response rate with IL-2 and a median progression-free survival of 13.8 months. So again, only 40 patients in this study, but a compelling compared to historical controls, and he is presently enrolling a randomized phase 2 study of this combination. This idea of combining these agents with immune checkpoint inhibitors has been looked at by many groups in the laboratory. Here on the left, we see a combination of mositinostat, another HDAC inhibitor, with PD-1 or PD-L1 antibodies in a lung cancer model, and showing that the only places where tumor regression was seen were seen in patients that got combination therapy. And the change in polarity as well as the change in cell number when it came to the tumor microenvironment was exactly in the direction that you would want, with decreasing regulatory T cells with mositinostat, whether given alone or in combination with PD-L1, and increases in CD8 positive cells as well. And then here on the left, again, Pam Munster's work looking in breast cancer, combination immunotherapy with CTLA-4 and PD-1 was given with entinostat, and here you see a very nice benefit in this animal model system when that triplet was given. That validated work that had come out of Shibin Zhao's lab a few years earlier, looking at combination CTLA-4 and PD-1 therapy with combination epigenetic therapy in CT26 and 4T1, so a highly aggressive breast cancer model and colon cancer model, where you can see nice regression of these tumors. In fact, these animals, majority of the animals were cured in combination therapy, and you see the metastasis had also decreased down here in this figure. Now, Shibin's group gave a relatively high dose of entinostat, and we were unsure whether that dose was a dose that was going to be tolerable, but in their hands, when they were looking at what happened at the tumor microenvironment level, the CD8 positive cells really didn't shift, but what they did see was a small decrease in regulatory T cells, but a substantial decrease in MDSCs in this initial cursory profiling that they had done in their paper to look for mechanism. So based on those data, Liz Jaffe and I moved forward in a collaboration interested in exploring this in pancreatic cancer as well. And this is Brian Christmas, who is a graduate student in the lab. This is his thesis work, which he had funded by a Lustgarten grant. And so we were lucky to be able to try to elucidate this a little bit better in pancreatic cancer as well. And we initially saw very discrepant results than what had been previously demonstrated by Shibin Zhao's group. We didn't see decreases in MDSCs. We instead saw increases in MDSCs when these animals were treated with entinostat. Further profiling of these myeloid-derived suppressor cells did show that there was a potential decrease in the MMDSC compartment, which arguably may be the more immunosuppressive cells, but the substantial number of cells that were MDSCs were far greater in terms of the number of GMDSCs that were present compared to MMDSCs. But then Brian moved on to try to assess their functional status, and that's where it got more interesting, because when he did co-culture assays with CD8 positive cells, entinostat treated cells in these animals were able to survive, and CD8 positive cells were able to propagate where they were completely suppressed in any arm that didn't contain entinostat. And arginase activity, which is also a surrogate of functional MDSCs, also decreased substantially both in the G and the MMDSC compartment. The functional studies of this combination in the animals also showed an improved, statistically significant improvement in survival for these animals. And based on these data, we have moved forward on a clinical trial in pancreatic cancer. There also is a cholangiocarcinoma cohort to this as well, because our phase 1 study actually had a cholangio response. But we've been very interested in the pancreatic model based on what was shown by Brian's work. This study has a lead-in period of 2 weeks of entinostat alone, with biopsies that are done at baseline and only after entinostat therapy, for us to try to isolate if we can. We don't know if that biopsy is a little too early, but we didn't think in pancreatic cancer we could get away with longer than 2 weeks of just entinostat therapy. So 2 weeks of entinostat with paired biopsies before and after, and then a third biopsy done a month later after the patients are started on nivolumab. This is unpublished data, but is the early results of our study. 22 evaluable patients have been enrolled in this study, and we have had 3 patients who have had partial responses to this combination, and none of those patients were microsatellite unstable. All 3 of those patients have had durable responses to therapy. We still have a patient who is presently on at over 1.5 years on study. And this patient, who was our first responder, you can see here as her lung lesion disappeared, she had an almost complete response to therapy with normalization of her CA19-9. And we actually lost this patient. She passed away, not because her cancer progressed, but a year after she came off of therapy and was just hanging out because she didn't want to come into Hopkins anymore to get treated. She decided to get surgery to repair a hernia and ended up having a septic complication of her hernia surgery. But all 3 of the patients had relatively durable responses that we've been excited about. That's still only a 14% response rate. And so trying to identify what about those patients made them be the patients that responded is really our next challenge. And we're hoping that our biopsies will help with that. And we're not the only people that are seeing responses with this HDAC plus PD-1 combination strategy. So this is Ryan Sullivan's abstract that was presented as an oral plenary session at AACR just a couple of months ago. He presented data looking at pre-treated melanoma. So these were patients that had previously been exposed to PD-1 therapy and progressed on PD-1 therapy that then went on to get combination therapy with entinostat PD-1. And their response rate, you can see here in the waterfall plot, they had multiple patients that did very well with RECIST criteria responses, a response rate of about 20%, and the spider plot shows the duration and durability of those responses too, with the median duration of response in those patients being 13 months. They have been trying to identify biomarkers, as have we. We've actually been working in concert with them, looking at our samples and their samples. They looked at circulating CD8 positive and MMDSCs and found that there did seem to be a decrease in MMDSCs in the periphery for patients that were responders, but there is nothing predictive at baseline in this patient population so far that was able to predict the 20% that did well and the added 20% that had stable disease. And in lung cancer presented earlier this year, same combination in lung cancer that had previously progressed on PD-1 therapy. Again, we see multiple patients that had durable responses to treatment with combination therapy, but a response rate of only 10%, though 50% of patients had stable disease. So again, another place we're trying to identify who these people are and how we find them is going to be key as we try to move these therapies forward. They had peripheral blood on these patients and looked at baseline monocyte levels. There did seem to be a trend towards high monocyte levels at baseline, with those patients potentially having a target present to decrease and being on trial longer with enhanced durability. But that was not statistically significant. And you can see there were plenty of patients that had high monocyte levels that didn't do well on the trial as well. And then the last working hypothesis that we're working on presently is the idea that it may be a component of MYC modulation that is important. Entinostat is known to affect MYC and decrease MYC expression, and MYC expression has been associated with resistance to PD-1 therapy. And so looking at 4 paired biopsies, so it wasn't very many biopsies, it's very hard to make anything out of it, but doing an unsupervised analysis of the 4 responders, what they found was in those patients, there was decreased in MYC when we did an they did an unsupervised analysis. And so that's just one working hypothesis, but clearly there's a lot of complexity to that as well. Now, before I end, I don't want to forget about the demethylating agents. There is a lot more clinical data that's present with the HDAC inhibitor combinations, but we remain interested in the idea of looking at demethylating agents as well. And Anthony Alcuri and I are co-running a clinical trial looking at guadocytidine in combination with durvalumab in patients with pancreas hepatocellular or cholangiocarcinoma. That trial is presently accruing, and we anticipate that it will finish accruing in the next year. But we hope that we'll see some benefit in that patient population or one of those patient populations as well. And we're about to report out on this clinical trial as well that was designed with NITA to look at whether we need an HDAC component. Do we need a demethylating agent, or is it the combination that might be most beneficial when we try to do immune modulation? So this was a clinical trial that was actually designed as a phase 0 study. So each one of these arms only has 5 to 8 patients in it and is designed with a primary endpoint of looking at tumor infiltrating lymphocytes to determine which of these combinations have the most impact on tumor infiltrating lymphocytes that then can be moved forward into a properly powered phase 2 study. And again, we expect that study to report out by later this year. I didn't want to close without mentioning all the other epigenetic agents that are out there that we do not have as much data regarding, and that the epigenetics community is just starting to work through. There are drugs in the clinic. There are specific and non-specific chromatin remodelers. There are epigenetic readers like bromodomain inhibitors, where there are hints that there may be immunomodulatory impact as well, but all very early preclinical work. And so these are areas where in the next 5 or 10 years, I think we in the epigenetics world are really interested in trying out some of these new agents to see what their impact is as well. So in conclusion, I hope that I've both given you a flavor for what is happening in the epigenetics modulation world, but also some enthusiasm for the fact that even though these drugs are so nonspecific, and when it comes to an era of science where we are trying to understand such detail about cancer biology, drugs like epigenetic drugs can be really dissatisfying, and we understand that because they're so broad in their effect. But maybe this breadth is actually what is beneficial when we are trying to work with complex systems like the immune system or chemoresistance. And so these drugs, I still believe, have significant potential if we figure out how to use them better and which patients we should use them in. But we need to make sure that our clinical trials are designed in such a way where we can really answer some of these questions with the 1 or 2 responders that happen on each of these studies, so we can learn better what is actually happening when we combine these drugs. And with that, I'm just going to thank the many people over the last 10 years. Nita, you came in a little late, but I already thanked you. But I'm going to thank you again for being a fantastic mentor and bringing me into this field. We always have to thank our patients when we talk about this kind of work. They have been through, patients and their families, through many negative clinical trials to just get to the point now where we might be able to benefit them. And we've had a fantastic research team, both at Hopkins and across the country. And we've even had a couple of international collaborators that have been really, really helpful. So with that, I will take any questions.
Thanks a lot for a wonderful talk. We have time for a few questions or comments. I'll start. Clearly, you know, there's activity. To see activity in IO refractory patients is huge. For the lung cancer, that group was here actually to talk to us recently. And why would monocytes have an effect? I guess it's hard enough to know what the negative regulators are for the IO response to begin with, and of course, what's the epigenetic therapy is modifying. Why would monocytes have a role?
So the interest in looking at the monocyte group was to try to subdivide with the MDSC story that was coming, to look in the periphery and to see whether there was a monocytic component to it. And from my conversations with them, it looked like they were looking at many different components, and that was the one that hit when they were looking at that analysis. But I think that's the challenge, is that we don't understand whether that's really the underlying story or not, and why why that would be the appropriate biomarker. And more, I think people are believing that what we need is, as Nita defined in her work, biomarker signatures that take into account multiple different aspects, multiple different immune cell subsets.
Or you'll get that from all your tissue pre and post studies.
We will, but we are, but the challenge, of course, is in the pancreas study, we have 3 responders. Luckily, those 3 responders have 3 biopsies each, but, and we have a few patients with stable disease, but when the numbers are small, even when you do these broad explorations, it's very hard to know how much of this is just accidental hits or not. So pooling, I think the ability to pool the data we have with the melanoma people, with the lung people, that's going to be where we might get bigger numbers that would be more informative.
Absolutely. Charlie?
Great talk. I'm curious because in your last slide, you mentioned it, the ability that maybe even looking specifically at chromatin remodeling as a means to modify or enhance the immune response. About 25% of cancers actually have mutations in that particular region, those genes. Is there any evidence that those mutations can drive response or resistance to immune therapies?
So there have been a few studies that have looked at, for example, EZH2 mutations and small cell lung cancer, and there hasn't been a great correlation between the agents that we presently have to use and the mutations that occur in the solid tumor setting. And then there's TET2 mutations where there has been some potential preclinical work that looks at those TET2 mutations, but it's like less than 1% of tumors that have that particular mutation. So I think they end up causing such widespread dysregulation that it's, once you've got that mutation, just reversing that one piece may not be enough.
Neil, thanks so much for being here and really presenting all the work you've done in this field. It has come a remarkable way. Let me ask you your comments on differences between demethylating and HDAC inhibitor. In the preclinical work, they seem to be almost comparable, yet clinically there seems to be a little bit of difference in signaling, especially in the immunomodulatory behavior. Can you comment a little bit more on that?
So I'm not sure I would agree with you. I don't think that the demethylating agents have been properly tested with the right drugs in combinations with immunomodulatory agents, whereas the HDAC inhibitors have been. And I think for a long time that was because of where those drugs were in their development. So these were more business strategies. Azacitidine and decitabine were already off patent, and so nobody was really interested in using those drugs. And so the guadicitabine study that we are talking about, that we have in GI cancers, there's a similar ovarian cancer study that's going to report out. I think those data are still developing, and we don't know if they're going to be as interesting or not. Because again, the response rate, even with the HDAC inhibitors, looks to be in that 10 to 20% of refractory patients. And so I just don't think we have the answer to that question yet.
Great. Any other questions? Seeing none, Neil, thank you for a wonderful talk, and we'll enjoy having you here the rest of the day. Thank you.